So mechanisms that encode graded outcomes rather than all or none binary responses are a fundamental feature of life. And one of the processes in which the right exact amount is extremely important is when it comes to motivation. So motivation is a neuronal representation of internal state that drives goal-directed behavior, so that these behaviors will be expressed at the right time, at the right amount, and at the right context. And my lab studies the interplay between social interaction and motivation, and the context for our studies is that living in a social environment involves diverse types of interactions during which animals integrate the environmental input together with their internal state, and subsequently they choose one action over another to increase their chances of survival and reproduction. And in this respect, we're interested in trying to understand how social interaction affects motivation, and this in turn changes, for instance, sensory sensitivity, the value of reward, whether the lack of reward could be stressful to animals, how does social interaction and motivation affect social group dynamics, and how also this affects changes in reproductive physiology. And we studied these questions in Drosophila melanogaster, and that is based on the notion that mechanisms that process motivation, reward, and also social interaction evolved early in evolution, and that there are similar, probably ancient building blocks, genes, and principles that govern them. So if we can understand some of these processes in flies, this could also tell us something about more complex systems. And flies have about 150,000 neurons, and we have tremendous amount of neurogenetic tools to manipulate their nervous system. So our approach to these questions are to take flies and expose them to different social conditions or experiences, for instance, being socially isolated, or to have group living, or to have different sexual experiences, and then we measure different motivated behaviors. So for instance, we look at how does social condition affect male courtship behavior towards females. Should I try that? Is it better now?
Is it fine?
Okay. And we look at also at other competitive behaviors to see how they're affected by prior experience. For instance, here you can see competitive behaviors or aggression between two males, where one male drops its entire weight on the other male. It's called lunging behavior. We also study social group dynamics and social networks, and the interplay between how prior experience and motivation affect the formation of groups. And looking at other motivational behaviors, so we use ethanol consumption, voluntary ethanol consumption, as a readout for the amount of motivation a fly has. And in this system, we could simply provide the flies with two choices between food and food that is spiked with 15% ethanol, and we can measure the amount of ethanol consumption. Other motivated responses that are associated with reward systems are trying to mimic the operant system by Olson-Milner, in which rats press a lever to stimulate, self-stimulate rewarding neurons. So flies, we don't have a way to teach them to press a lever, but we have two systems that allow us to have some kind of an approximate system for that. And we use optogenetics instead of electrical stimulation. And in this case, we have flies that express channel rhodopsin within neurons that promote reward. And they are giving a chance between being in a part of the arena in which their neurons are illuminated and optogenetically activated versus the non-illuminated zone. And you can see here in this movie that flies can choose to remain within a zone that leads to optogenetic activation of their neurons. Another system that we are currently using a lot is an actual operant system that is also based on optogenetics, having the channel rhodopsin expressed in reward neurons. And in this case, flies can choose between two electrodes. When they, for instance, touch the left electrode, this activates the optogenetic or the LED light. And you can see here that in every of these drawers, there is one fly that stimulates its own neurons, leading to pleasurable responses. In terms of mechanism, the other layer that we're studying is how is the information that is gained from the interaction is encoded within the brain. So again, exposing flies to different conditions, and at the end we look at motivated behavior, but in the middle we're looking at certain neurons and their molecular profile, and the way we do that is that we express a GFP that is bound to the membrane of selected neurons, and then we could dissociate the brains, immunoprecipitate specifically subsets of neurons. It could be 6 neurons, 20 neurons, 100 neurons, and then profile them using RNA sequencing. And eventually what we try to do is to connect functionally between the input, changes in molecular modifications within subsets of neurons, and how does this modify behavior. So today I'll try to give you several examples of the studies that we're currently doing. So I'll try to go through our flies. Social, do they generate complex social interaction in groups? How does sexual experience modulate their behavior? And I'll end with a more molecular project bridging the gap between RNA editing and behavior. So we wanted to ask, how does group behavior in Drosophila unfolds under different conditions, asking whether flies do generate groups with different structures under various conditions. Is the group affected by the properties of its members, and what are the some of the underlying neurobiological mechanisms? So we search for conditions that can facilitate the formation of distinct group behaviors. We thought that groups that have flies with different social histories will have different emergent group structures. So we took very extreme conditions. On one hand, flies that from their eclosion were kept solitary, and flies that were socially raised, thinking that the group signature of socially raised flies will reflect some kind of a snapshot of already established relationship between members of the group that developed over the course of housing, whereas in solitary flies, when we put them in groups and assay their behavior, we'll see a signature of what reflects an initial interaction between members of the group for the first time. So our setup is based on putting the flies in circular arenas, recording their behavior using tracking algorithms to track their position, and from that we extract both kinetic features and also use machine learning algorithms that were developed at Janelia to classify their behavior into 8 complex behaviors, and also calculate from their location and approaches an interaction network. So analyzing the groups of these 2 distinct experiences, what we do see that flies that are socially raised, in terms of activity, they have reduced activity compared to solitary flies when interacting in a group. So they have reduced velocity, they exhibit less walk and less turn. And looking at various social behaviors, so what we see is that flies that are socially raised have certain reduction in some of their social behaviors, so they have reduced close touch behavior when one fly touches the other on the legs or wings. They exhibit less approach to one another. They chase one another to lesser extent, but they have a behavior in which within several minutes from the beginning of the experiment, they cluster into groups and sit together for long periods of time in a quiescent state. And these differences between the two types of conditions are not just a matter of arousal level. So if you take single flies from this group and assay various features of their behavior, we don't see any difference. So this means that the social experience effects on behavior are an emergent property that is expressed only during group interaction. So now I'll switch to telling you a little bit about social networks. So for an interaction to be registered as an interaction, we require the distance between two flies to be less than 8 millimeters, that we measure the angle in which fly looks or perceives other flies, and it has to be more than 1, and the length of interaction more than 2 seconds. And we look at different features of their networks, and that is the strength of individuals within their network, how dense are the networks, are they highly dense or sparser, if the networks have modules within them, so are they divided into subgroups, and what is the variation in the strength of individuals within the group, and we measure that using either the number of interaction or durations of interactions. So comparing between the two groups, what we saw is that groups that are composed of socially raised flies have higher modularity, and also the variance of the strength between flies is higher, so having more complex groups that have subdivisions. And when we look at the number of interactions, so we could see that groups of solitary flies are much more dense, but still the modularity is lower. Another aspect that we kind of felt by looking at the behavior, it seemed that the behavior of the socially raised flies is more variable. So to see if that is actually more than intuition, we calculated the variability of the different features, either looking between flies, so this is some kind of a measure or proxy for personality, between groups of flies, so this could be a proxy maybe of culture, and between flies of the same group, so this is simulating hierarchy, and measuring that for all of the different features by the 3 different measurements, looking at the number of features that show variability between the groups, we see that socially raised flies are much more variable in their responses, and this tells us maybe that their behavior is more diverse than that of solitary flies, and this possibly reflects a broader repertoire of behaviors that they engage in. So having about 60 different features, it's quite hard to look at each of them separately. So what we do is we normalize everything by z-scoring, and then use scatter plots to look at patterns of these behaviors. So again, you can see between the two groups that solitary flies are more active, they are more have more social interaction, and that the socially raised flies have higher features of social clustering. So which sensory modalities are responsible for these emergent differences? So we started with very simple experiments in which we looked at visual sensory information and olfaction. And in this experiment, what we did is we tested the flies of the same groups or socially raised groups flies under 2 conditions, under light and under dark. And you could see that switching to testing the flies in dark induces a pattern that is more similar to solitary flies. So visual cues are important for the properties of the pattern of socially raised flies. In terms of olfaction, we manipulated neurons that perceive pheromone responses that are specific for males. So males have a specific pheromone which is called cis-lothianyl acetate, or CVA, and there are 2 subsets of neurons that perceive these pheromone, so we could manipulate those during the housing condition and test the effects. So here are manipulation of one set of neurons which perceive the pheromone for long term, and these are OR65A, and these are neurons that perceive CVA acutely. So you could see that in the orange dots, these are experimental flies inhibiting the ability of flies to sense a male pheromone during the social raising induces a behavior which is more similar to solitary flies. So far, we've used the homogeneous groups of flies. Next, we asked what happens if we look at more complex situation in which we have flies that have different social experiences. And in this case, what we used is flies which are hyper-aggressive. This is by a specific mutation. And we mixed, during the test, the groups. So these are 10 wild-type flies. Here we have 1 mutant fly, 2, 3, 4, and 5. And we analyzed the behavior. And since we have a large data set, we clustered them to see if there are interesting patterns. So what we see, so here is from 100% wild type to 50% wild type. So you can see that we see certain features that are induced with this mixing experiment and certain features that are reduced. And it could be argued that what we see is some kind of an average between the two groups rather than a more complex pattern. So to see if this is correct, what we did is we looked at the distribution of the flies. So in this case, just as a proof of principle, we used TSNE to see if we could subdivide the groups of flies that were tested in the light and in the dark, and you can see great separation. In the case of this experiment, so you can see that the distribution doesn't allow us to know exactly which group is what, meaning that the heterogeneous groups of flies have an emergent structure that is more than just the sum of the parts. So now I would like to talk about how sexual experience affect male flies. So previously during my postdoc, I exposed male flies to 2 extreme sexual experiences. On one hand, successful mating events with plenty of virgin females. On the other extreme, males that are repeatedly rejected by mated females that are not receptive. And what we discovered is that these experiences affect their motivation to consume ethanol as an alternative reward. So rejected male flies have higher preference to consume ethanol, and this is causally mediated by neurons that express the fly homologue of neuropeptide Y. That serves as an internal representation of the level of reward. So to start with mating, what I forgot mentioning is that we've shown that successful copulation is rewarding to male flies. What exactly is rewarding in mating? So we asked if this is just being with a female pheromone, courting after females. And eventually we thought what might be obvious, that in order for the experience to be rewarding, males have to complete the entire process and that ends with ejaculation, so transfer of sperm and seminal fluid. So to test if this is correct, what we did is we optogenetically activated a subset of neurons, a dimorphic set of neurons that are male-specific, that are in the ventral nerve cord of the fly. When you optogenetically activate these neurons project to a male reproductive system, and this induces ejaculation in an artificial manner. So we asked whether we could take male flies without females and mimic completely all of the features of reward. So this is, would flies have a preference towards the zone that activates these neurons? Can we assay associative memory? Do we have induction of neuropeptide F, the fly homologue of neuropeptide Y, that is elevated in response to mating? And also, could this inhibit ethanol consumption? And I can say shortly, because this is already published, that we could mimic completely reward by inducing ejaculation in male flies, and asking how exactly is the information gets from the reproductive system back to the brain affecting NPF. One could argue that we have some kind of ascending neurons from the ventral nerve cord, the ones that are in charge of ejaculation, and they send their projection to NPF neurons, or that this is somehow sensed by mechanosensory neurons at the reproductive system of the flies. So in this experiment, what we've done, you can see here a movie in which male flies that lack the reproductive system are still courting, and we tested whether the optogenetic activation of the neurons that induce ejaculation are rewarding, and testing if they have a preference to the activation zone, and they don't, meaning that so that it's not directly from these neurons, but somehow sensed some kind of contraction within the reproductive system. So to follow that up, what we've done is we looked for varieties of mechanosensory neurons that are either throughout the covering the testis, the seminal vesicles, the ejaculatory duct, or bulb. And there are various mechanosensory neurons that cover those. So we used genetic drivers to manipulate these neurons. First, we looked at where are those expressed, and I really love this picture. It has a lot of beautiful images, but the bottom line is that these various mechanosensory neurons are expressed in different structures of the reproductive system, so this didn't help us to narrow down the relevant neurons. So then Scheer did a functional screen using the positional preference, asking activation of which of these neurons lead to preference towards the activation zone, and none of these led to preference. She focused on 2 or 3 subsets of neurons that express the mechanosensory receptor piezo, painless, and OMP-C. They have positive responses. Then she narrowed it down by looking at ethanol consumption and molecular changes, and that is NPF. And the only one that gave good responses are piezo. So they have positional preference to the activating zone. Their activation leads to reduction in ethanol consumption and also induces transcript level of NPF. You can see here by expressing fluorescent protein at the dendrites of these neurons that these piezo neurons have dendrites covering the accessory glands and the ejaculatory duct. One of the problems of these experiments is that these piezo neurons are expressed or are localized in many different structures throughout the fly organs, so we needed to narrow down their expression pattern using neurogenetic tools to have a more precise expression of only small subsets of neurons. And when we do that, so reducing the expression pattern, we still see beautiful expression within the reproductive system. And some of these neurons are also dimorphic, so they express the fly fruitless marker. And this is, again, their expression in the reproductive system. And some of these piezo fruitless neurons have their projection back or they are sent to the brain, and these are they're in close proximity to NPF neurons, so they're good candidates. So again, what you can see in this slide is that this is activating the entire population of piezo positive neurons. This gives a positional preference to a certain extent, but when we clean this driver up and having a very small amount of 20 neurons, we can see much stronger response in terms of positional preference and also ethanol consumption. So this is mating. I'll switch to talk about a little bit about sexual rejection. So as I mentioned a few slides ago, there is an inverse correlation between mating status and ethanol consumption. So males that have successfully mated have aversion to ethanol, while males that are sexually deprived have higher preference to ethanol. And this is inversely correlated with the levels of neuropeptide F in their brain. And as I mentioned, these are measuring ethanol as a proxy for the reward or for their motivation. So we looked for, and this could be argued that ethanol has other benefits, maybe it's calories, it's not necessarily motivation or reward. So we needed a more direct measure of the fly's motivational state. So in this system, the optoped system, flies are inserted, each of them into arena. They can choose to touch one of the electrodes. These flies have the channel rhodopsin expressed in their NPF neurons. When you activate NPF neurons, this is rewarding to male flies, and we look at how fast do they learn that this electrode is the one that activates the neurons. So these are without any sexual experiences. You can see here in this experiment, this electrode leads to optogenetic activation, where in this experiment none of the electrode does. And we look at their preference towards, so one is complete lever pressing or touches at the activating electrode, so this is preference index. So whenever none of the electrodes leads to optogenetic activation, they don't have preference. When one of them, the left one, induces optogenetic activation, flies do learn to do that. So now what about sexual experiences? So we compared flies that are sexually deprived or sexually rejected to male flies that don't have any sexual experiences. And what we see here is that flies that are sexually deprived, rejected by mated females, their learning curve is faster than naive male flies. So this tells us that sexual rejection is a reward deprivation that induces high motivational state. How does this experience affect interaction with females? So interaction with mated females is long known to induce what is known as courtship suppression. When a male interacts with a non-receptive female, it learns to associate female behavior and its pheromones, and to, in the next encounter, to choose a more receptive female or to reduce its actions toward mated females. In this experiment, Yulia did manual scoring of many of the different behavioral features that males exhibit during courtship, so measuring their wing vibration. They use wing vibration to sing a song, a love song to the female. She looked at the number of licking events, so there is a licking event between copulation. How long does it take them to start courting? And what we see is that indeed, as known, we do see reduction in the time these males spend courting, but when looking at many different other features, these rejected males, their wing vibration, number of licking, how long does it take them to try and copulate, these seem similar between the two groups. And in some cases also, we see that these rejected males also invest more time in the bout length of copulation attempt. And this tells us that although it is thought that there is a courtship reduction, we actually see that sexually deprived male flies exhibit persistent motivation to obtain a mating reward. How does this affect, this experience of being repeatedly rejected affect male-male interaction? So in this case, we use the same paradigm of 4 days of exposing the flies either to successfully mating events or to sexual rejection, and we assayed their interaction in groups. And you could see that rejected flies are much more active in these many different activity-related features. They have reduced social interaction, and they exhibit reduced social clustering. When we looked at their social networks, you would expect from flies that are more active to have more chances of interacting with other flies, but actually their networks are much sparser than that of mated and naive, and this suggests that male flies that are rejected are actually avoiding interaction with one another, and we could see that support for that idea by looking at the distances between flies, whereas mated males become closer to one another along the course of the experiment. Rejected males maintain the same distance between one another. In terms of competitive behavior, when we so before getting into competitive behavior, one of my colleagues who is an ecologist told me that he thinks that rejected males, whenever they meet a mated female, this tells them something about the social environment in which they live. So if every female they encounter is already mated, and I have to say that we do these mating experiences, these male flies do not see any other male until we put them in groups. So the conditioning or the experience phase is single males with different types of females. So whenever they encounter a mated female, that tells them that there is a lot of competition around because all the females are mated. So if this is correct, we would have expected that these rejected males that have high motivation to mate to maybe have increased competitive responses. So in this case, we measure their aggression behaviors in couples towards one another. So two couples of rejected males compared to couples of mated males. Rejected males are much more active compared to mated males. And when we put them in mixes, so mated male versus rejected males, and we measure their behavior, so rejected males exhibit higher aggressive or number of lunges towards the mated ones, so the more aggressive. So this is quantifying this graph. Looking at their mating response, if they do get a chance to mate, what these rejected males do is that they increase their investment in mating. They mate for a longer time, for about 3.5 minutes longer than males that didn't undergo rejection. And they also change the composition of transcripts of proteins that males transfer to females. So in this case, they're regulating proteins that have the capacity to change the physiology of the female. One of these proteins, it's called sex peptide, and it induces post-mating response in flies, reducing the tendency of females to mate. So we tested what happened to females that mated either with naive males or rejected males, and we scored the number of males that are courting these females. And as you can see, females that mated with rejected males, less males or reduced number of males are courting these females, so they're less attractive. But also these females, they're remate in a lesser extent than females that mated with virgin males. So again, suggesting that the fact that they change their physiology of proteins and also their mating duration might contribute to their competitive response. But is there a cost to this persistence, motivation to mate, and increase in competitive response? So Yulia looked for measures that might suggest that there is some kind of a cost for that. And what she documented actually by accident is that whenever these males are rejected, they have increase in grooming behavior, which is a well-known behavior in mammals that is considered a displacement behavior. So this led us to test whether they're more sensitive to stress, or does it affect their stress response. And what she did is she tested their sensitivity to either starvation or oxidative stress, and in both cases, rejected males are much more sensitive to both starvation and oxidative stress than mated males or males that don't have any sexual experience. And this tells us that sexual rejection compromises their ability to cope with stressors, and this might be first evidence in flies that you could have induction of social stress in flies. So trying to understand the molecular mechanism, what we did is we took these rejected males, mated males, and also naive males, and we profiled several subsets of their neurons. We profiled neurons that have the receptor for NPF, neurons that have octopamine, which is the fly neuroadrenaline, and serotonergic neurons. And you can see here that we do get interesting patterns of certain genes that are upregulated or downregulated, each of these neurons in response to either mating, rejection, or no sexual experience. Trying to understand some meaningful pattern from, or some causality between all of these different changes. So we looked at what molecular changes are unique to rejected males in neurons that have the receptor for NPF. And what we see is that there is an induction of a ubiquitin ligase protein and several genes that are related to the insulin FOXO stress pathway. So to identify a causal link between, for instance, the regulation of UBC7 and rejection, what Yulia did is she used CRISPR, tissue-specific CRISPR, to specifically induce a deletion within UBC7 at NPF receptor neurons and assay their behavior. So male flies that lack UBC7 at NPF receptor neurons, their motivation to court is higher. The time it takes them to start courtship is shorter, and the time to copulation is also shorter, and this is the latency to first copulation attempt, and this is the latency to copulation. So this is well aligned with the overall behavior that we do see in these flies. But one could argue that the cost of having being more sensitive to starvation or to oxidative stress is a matter of metabolic responses. That male flies that have high arousal level, that are trying to compete over mating, have shortage of metabolic of metabolites. So we first compare their weight, looked at their lipid content, also glucose levels, both in their body and in their hemolymph, which is the blood, and we didn't see any differences that could explain that. We did a more elaborate metabolomic analysis, and we do see some differences, but in overall they can't explain the phenotype. So this led Yulia to think that what we know already is that rejection leads to disinhibition of the neurons that are beneath or one synapse down to NPF, and these are the NPF receptor neurons. So she thought that what happens is that in rejection, these NPF receptor neurons are more active, and this could lead to sensitivity to stress. So to test that, what she did is she knocked down the receptor for NPF in these NPF receptor neurons and exposed the flies to the different conditions. So here are the controls where there is no reduction of the receptor. The rejected males are more sensitive. Knocking down the receptor, they become as sensitive as the mated. So taking this one step forward, we mimicked rejection by activating those neurons. Activating optogenetically NPF receptor neurons lead to sensitivity, and I'll skip that. This is just showing that this activation of these neurons require the release of dense coarse vesicles. And then she narrowed down from 100 neurons that are NPF receptor neurons to smaller subsets of neurons. One of the subsets which is relevant are the 12 neurons that co-express the neuropeptide tachykinin, also known in mammals to affect arousal level and aggression. And when activating this small subset of NPF receptor tachykinin neurons, she could mimic the effects on sensitivity, but also some of the effects of their social responses. Okay. Do we still have time for me to talk about RNA editing? Okay, cool. So now I'm doing like a sharp switch to talk about RNA editing. One of the reasons I'm studying RNA editing is when I joined the life science faculty at Bar-Ilan. Erez Lebanon, who is one of the leading researchers in RNA editing, approached me and said, I think that RNA editing could be a mechanism that encodes information. So I'll tell you a little bit about RNA editing. I know that this is quite molecular, but I'll try to stay more broad. So the basic dogma in biology is that we have DNA, and then from there messenger RNA that gives rise to protein. And there is an enzyme called ADAR, which is capable of taking certain adenosines within the sequence and converting them into inosines. Inosines are recognized by the molecular machinery as guanosines. So if this happens in coding regions of proteins, there is a change of amino acid that could eventually lead to change of the physiology or the biochemistry of the protein. One of the interesting things about RNA editing that it happens to a higher extent in brain tissues. So here what we did is we profiled RNA editing within different neuronal populations in the fly brain, using again the same approach of isolating different neuronal populations. And the bottom line of this study is that each subset of neurons within the fly brain have a different repertoire of RNA editing. And I'll give you just one example of how strong can be the RNA editing can be the diversifier of the proteome. This is, for instance, the fly sodium channel, which is called paralytic. And it has 10 different editing sites that are differentially edited between different subsets of neurons. So, for instance, in neurons that are called corazonin, they have about 60% more editing of a specific tyrosine into cysteine compared to the average neuronal population. And in the different editing sites, there is a recoding in NPF receptor neurons, which have 45% more editing of a specific isoleucine to valine in these subsets of neurons. And so each subset of neurons have a different combination of RNA editing levels only of the sodium channel. So this could give rise to tremendous variation in the composition of sodium channels within subsets of neurons. So what does this, for instance, tyrosine does? So this tyrosine is located within the voltage sensing domain of the sodium channel. And when we model this localization and converting this tyrosine into cysteine, so this recoding is expected to eliminate this bulky occlusion in the gating charges of the channel, and this might affect the physiology of the channel. To get a sense if this is true, what we did is, so this tyrosine is highly conserved from flies to humans. So in human sodium channel, there is a tyrosine. So what we did is we mutated the human sodium channel, converted this tyrosine into cysteine, expressed it in cells, in 293 cells, and in collaboration with Alon Condon, we started measuring their voltage sensitivity. So this is in red is the wild type channel, and in blue is the mutated channel. And you can see that replacing this tyrosine into cysteine makes this sodium channel less sensitive to changes in membrane potential. And this is just preliminary, but there are other evidences suggesting from other studies that you could change the physiology of the channels just by recoding certain amino acids using RNA editing. So in this study, what we did is we took a certain channel in which the RNA editing is conserved in all species of Drosophila. Some of them are separated millions of years from one another. So we focused on one of them, which is RNA editing within a glutamate-gated chloride channel, which is called glucyl alpha. It has 6 editing sites. One of them is really conserved. It's an isoleucine within the ligand binding domain. So glucyl alpha is expressed in the olfactory system of flies. It is expressed in projection neurons and also local neurons that modulate the olfactory responses. What we did is we used CRISPR to generate flies that are constitutively unedited. So in all of the isoforms of the channel, this is adenosine, so there is always isoleucine. And we assay different, so this is just to show that only this site and the one close to it is affected by this CRISPR modulation. The other site is non-recoded, so it doesn't affect the function, and the level of the protein is the same. Assaying their behavior, so we use a very simple olfactory responses, that is what is called olfactory trap. Flies can choose between to sense and get into olfactory trap towards apple juice or ethanol. These are the wild type responses, and these flies are unedited flies. You can see that they have reduced preference to apple juice and ethanol, which are appetitive. And again, in a different assay in which we look at how fast they reach the odor, again, a reduction in their appetitive response towards apple juice and ethanol. And in this system, in collaboration with the lab of Chico Parnas, we could look at the fly's trajectory in channels in which they get the odor from either this side or this side. So flies, whenever they are exposed to an aversive odor, they go to the other side. And in this case, what you can see, both acetic acid and 2-butanone are aversive orders. So you can see aversion of the wild type controls, whereas the unedited forms of flies that have unedited glucyl alpha are not averse. Also, another behavior which is olfactory dependent is egg laying in females. So female flies, when they do get a chance to lay their eggs, they rather lay their eggs on food that have acetic acid. This gives, provides protection from parasites for their progeny. But in terms of olfaction, they are averse from this smell. So there is a conflict between the place they want to be and the place they want to lay their eggs. So you can see that they have an aversion to acetic acid in terms of their position, but they lay their eggs on the side of the acetic acid. So when we use this assay to look at another olfactory response, you can see that wild type flies, females have an aversion to being in the acetic acid side versus the glucyl alpha unedited flies. And looking at oviposition, so these females lay most of their eggs on the acetic acid side. I'll say shortly also that this manipulation of unediting of glucyl alpha affects female receptivity to male courtship behavior and also affects male aggression. So males that have unedited version of glucyl alpha have reduced number of flanges of aggression, and also it takes them longer time to start the aggressive response. What we did next is we wanted to rescue this behavior by expressing in central subsets of neurons, in projection neurons within the olfactory process, to express the edited form of glucyl alpha. So what you can see here, these are the genetic controls that unedited flies that don't have the preference. And when we rescue glucyl alpha specifically in the projection neurons, we see rescue. This is also true for copulation and also for aggression that could be rescued. In terms of calcium imaging, so this is in collaboration with the lab of Chico Parnas. When we analyze the calcium responses to different odors within the entire glomeruli, the olfactory bulb of flies, we didn't see any differences between the two lines. So we went into analyzing specific subsets or specific glomeruli, and specifically the one which is called VA1V. We could see strong differences between the unedited flies and the edited controls. So suggesting overall that RNA editing affects the responses of flies to different odors, and that this happens within olfactory processing neurons. And I would like to finish with just 2 slides about showing where we're going. So one of the projects and approaches that I'm really excited from, which for me as a molecular biology are some kind of a fiction science. And this is the collaboration we have with Shachar Alon at the engineering at Bar-Ilan University. And it is whenever we analyze the transcript of neurons, we always take into account that, that we just look at what is expressed in terms of the entire transcripts. But neurons, thinking about their elaborate structure, one can think that information is not only about how many reads of RNA per gene there is, but also where it is localized. And having a complex structure, one needs the ability to look at specific RNAs within the brain. So Shachar Alon, during his postdoc at MIT, developed an amazing technology which is called expansion sequencing. The idea behind that is you take the brain, you expand it enough to be able to have high enough resolution to discriminate between 2 points in the brain, and then we perform RNA sequencing specifically within the tissue. So what you can see here is, so we have probes for 300 genes. Each gene has 10 different probes, and so the RNA is fixed and then the brain is enlarged, and we increase the number of transcripts by PCR within the tissue, and then sequence by adding the nucleotides, which each of them is fluorescent, and each of these dots in space is actually a specific transcript. So here you can see a subset of NPF receptor neurons, NPF neurons, in which we express a fluorescent marker in the membrane, and each of these dots within the brain is a different transcript, and nowadays we're trying to get a complete understanding of what is exactly expressed in different subsets of NPF neurons in the dendrites and synapses, and I think that adding this information is going to really promote the field forward. And with that, I will end, and thank you for listening. So discriminating between mated female and virgin female is based on experience. Yeah, it's based. No, it's pheromonal experience. So whenever they court a female that is already mated, this female has a certain pheromone signature, and they generate an associative memory. The next time they realize or they are exposed to the same signature, they reduce their coaching.
But this is like a female-specific pheromone?
No, this is a male pheromone that is that males transfer to females. So mated females have a certain smell that is different than virgin females. But they also are capable of distinguishing between all females, young females, immature females, to invest their efforts on the right target. Yes.
Great talk. I'm wondering how specific NPF is to the social experience. Like, I don't know if there's any other paradigms where you can kind of frustrate the reward-seeking attempts of a fly and see if that modulates the NPF levels. Like,
so if another type of reward can modulate NPF levels,
yeah,
which is not social.
Which is
yeah. So exposure to ethanol induces rewarding levels of ethanol induces NPF to a similar extent than mating.
And then the NPF receptor, like how much does that? I missed this, but how much does that overlap with fruitless? Which I know.
Only subsets of NPF receptor neurons are fruitless positive. Actually, there is some overlap between NPF and NPF receptor. There's some kind of autoregulation. Two of these neurons are fruitless neurons, and there are male-specific as well. Yes?
Maybe it's a little bit technical, but when you write down the connectivity graph, the social interaction, so you showed us minimum requirement of the flies being able to see each other. But how do you construct the graph to say we need two flies to see each other and have engaged in certain behavior simultaneously?
So some of the analysis we've done is based on classifiers that classify if they're an approach, but you're asking about the network.
Yeah.
So all of our data is based on per fly per frame. In each frame, we look at one fly and we look at all of the flies. So in each frame, we ask if this male fly is in interaction with another fly. So we generate an interaction matrix.
So it's a directed, because you only look at one fly.
Yeah, so we did ask if we could see differences, if one is the one starting the interaction or the other, and we eventually realized that undirected, because if there is a small, like really short amount of time in which this is directed. Once the other one is engaged, it's also interaction.
So you have some requirement for being engaged. Two flies, you do it in the same activity, and they go in the line of sight of each other.
Yeah.
So a question about the first part. Sorry, maybe you mentioned it. When you looked at heterogeneous groups, you mentioned specifically high aggressiveness, but before that, when you were looking at those flies that were raised in isolation versus with social interactions, did you look at what happens when you combine those types?
So we didn't do that. We thought of like that. I was raised up as a biologist, as a chemist, and sometimes to be able to see something, we push things to extremes. So we thought that it would be more extreme. So I didn't mention that solitary flies are more aggressive. So if you raise flies in group, they learn to reduce their aggression. So to push it stronger into isolation, we use flies that are genetically aggressive, mimicking the isolated ones. But it's not exactly. I do agree that we should mix the. One of the limitations of this experiment is that the flies are not marked, and we don't know exactly which is which. So also when we try to discriminate per fly, if we could know which is the aggressive fly, and there is no, like, they really do not seem to be different in terms of like individual behavior. It's more of the emergent pattern.
I don't know much about flies, but those that were raised with social interaction,
Those that
were raised
Yeah.
with social interaction, does it matter if you then test their interaction with flies that they were raised with or with other
Great
flies?
question. I omitted this from. So one of the things I told Ilacha today about is that it is always interesting to ask if flies have individual recognition. If they know that you're Aya, and not just a person. And there is evidence that they do. And we thought that this is a great way of like looking into that, whether they engage differently with flies that they grew with or not, and we got very disappointing results. That it didn't affect that the structures were really similar. And we were disappointed from that. So this is not the right or probably paradigm to ask that.
Any more questions? Okay, great. Thank you very much.